Creating chemical reactions in the lab is almost like cooking in a kitchen. For example, making an exquisite tomato sauce for pasta requires mixing ingredients in a pan such as garlic, tomato, salt, pepper, and basil. However, there are different mixing options. You can start with onions, garlic, then the tomatoes, then salt, pepper, and finally the basil. Alternatively, you can try salt and pepper first, then the basil, and finally tomatoes, onions, and the garlic. The sequence of the ingredients influences the results. Chemistry is similar. If the 6 food ingredients were instead different chemical ingredients, that is, molecules that can bind together in a chain, then mixing them in different orders will produce different chains. And if you had to make a chain that is 6 molecules long, and all 6 different molecules are in different positions, then you could make 6 times 6 times 6 times 6 times 6 times 6, a total of 46,656 different chains. This would be very difficult to do in a lab and would require large quantities of materials, human resources, and lots of time. Researchers from several universities in Denmark have now collaborated in developing a new way of carrying out chemical experiments that use less of all these 3 types of resources. Instead of performing the experiments in large flasks, one at a time, the researchers have developed lipidic nanocontainers for carrying out the tests. In many ways, these containers are like tiny soap bubbles that are equivalent to an empty glass flask in which the researchers can carry out standalone experiments. The containers shown here are called target liposomes, which are immobilized on a fixed surface and are so tiny that more than 40,000 can fit in a square millimeter. The materials for the experiments are also encapsulated in lipidic nanocontainers called cargo liposomes. But how can you control the mixing of so many experiments at once? The answer is in the surface of the lipidic nanocontainers, on which the researchers tethered several strands of DNA. DNA strands with 6 different sequences are tethered to the target liposomes, and DNA strands are tethered to each cargo liposome that can only bind to one of the target liposome's DNA strands. These are called complementary strands. When the cargo liposome hits the right strand in the target liposome, the strands will intertwine like a zipper. The instant this happens, the cargo liposome and the target liposome will fuse exactly like 2 soap bubbles meeting, and the contents of the cargo liposome will be emptied into the target liposome. The target liposomes are designed such that several DNA strands are tethered on the outside so that more cargo can be mixed into them, with 2 different molecules now fused in the target liposome. When the researchers repeatedly add a mixture of all the 6 different cargo liposomes across the surface of the 40,000 plus target liposomes, the materials will be mixed together in the target liposomes in completely random order, which in many ways is like mixing chemicals together in a flask.
So the beautiful thing is that in this way we can make several thousand experiments at the same time, and all in an area that is not larger than 1 square millimeter. Each target liposome is its own little experiment.
Mixing the materials randomly is one thing, but how can the researchers identify how they were mixed? To solve this, the researchers developed a method in which each cargo liposome contains lipids to which various fluorescent markers are attached. These are in different combinations of red, blue, and green, and each cargo liposome thus emits light in a unique combination and quantity. Artificial intelligence can then interpret 40,000 individual light signals from each target liposome in seconds from images created by a special fluorescence microscope. The researchers can thus determine very precisely the order of mixing in each target liposome. The research group thinks that the new technology has great potential to save time and resources.
Among other things, one could imagine that scientists would put building blocks together for use in various contexts. For instance, the development of new medication.
You can read more about this research at sciencenews.dk.